Dear Chairman, dear audience, the therapy of patients who are suffering from a local or a systemic inflammation means an everyday problem at clinical departments. Among other things, the ischemia, red perfusion, and the regional ischemia cause an inflammatory response too. The ischemia means a total oxygen block of a system or a local tissue. And the tissue damage arises after the reoxygenization, like in case of transplantations, or right after the ischemia because of the oxidative and nitrosative stress. The systemic hypoxia is such a hardly treated like the previous problems. It can be induced by acute respiratory distress syndrome or chronic obstructive pulmonary disease or by emphysema. And the tissue damage is also a frequent consequence of systemic hypoxia, result of hypoxic stress response. Due to the hypoxia, the arterial oxygen tension decreases, whereupon microcirculatory dysfunction occurs. This leads to hypoxia of tissues and acidosis. Because of the lack of oxygen and acidotic circumstances, mitochondrial dysfunction causes hypoxic stress response associated with free radical production. Pathways of free radical production under hypoxic circumstances have an extensive range. Due to the hypoxia, xanthine dehydrogenase enzyme proteolyzes to xanthine oxidase, and the xanthine oxidase convert hypoxanthine to xanthine while forms reactive oxygen species like superoxide radical. The other component of this stress reaction, the nitric oxide. It can be produced endogenously by the nitric oxide synthase enzyme, but there is another way of the nitric oxide production under pH 7 by the nitrate reductase activity of the xanthine oxidase. Both sides of this enzyme is able to reduct the nitrate and nitrite to nitric oxide. So the nitric oxide forms a more reactive, more effective agent with superoxide, the peroxynitrite. The peroxynitrite takes part in membrane lipid peroxidation, breaks the DNA, and with aromatic amino acid nitration cause structural and functional protein modifications. So, all in all, the hypoxic stress response consequence of structural and functional tissue and cell damage. But how can we moderate this stress reaction? Our answer is a methane as a promising possibility. Here in this picture, we can see the ischemia reperfusion induced tissue damage in the small intestinal mucosa, as the previous results have shown at our institute. And here, the reduced tissue damage with methane treatment compared with the sham-operated tissue. Based on the previously demonstrated results, main questions is that the nitrosative and/or the oxidative stress response stands behind the process of the hypoxia. And the methane have a beneficial effect under hypoxic circumstances, able to decrease the tissue damage. To find out the answers, or aim to examine the effect of methane inhalation in systemic hypoxia in vivo, on the gastrointestinal system, on the lung, and furthermore to examine the signs of oxidative and nitrosative stress response. Our experiments were performed by SPRD anesthetized male rats. After the anesthesia, tracheostomy was performed to facilitate the spontaneous breathing and the respiration. And then the right jugular vein was cannulated for the infusion, and the common carotid artery was cannulated to monitoring the hemodynamic parameters and to 2 blood gas samples. After the preparation, mechanical ventilation was started with a rodent respirator with normoxic air for 30 minutes. Then the rats were randomly assigned to 5 groups, and the respiration was continued with different gas mixtures for another 30 minutes. The gas mixture of the first group contained normoxic air. The second and the third group were ventilated with oxygen-depleted air, contained 18% oxygen with or without methane. and the fourth and fifth group get on a stronger hypoxic gas mixture with or without methane in it. And at the end of the respiration procedure, we took tissue and blood gas samples. From the arterial samples, we measured the blood gas parameters with immediately post-sampling. And from the lung and intestine tissues, we measured the xanthine oxidoreductase kinetic activity with fluorometry, the nitrate-nitrate content, the nitrotyrosine level, and the myeloperoxidase activity. Arterial blood gases have shown that the methane did not exert significant effect on the arterial oxygen tension compared with the hypoxic groups. The xanthine oxidase enzyme is responsible for a big amount of reactive oxygen species under hypoxic circumstances, so we measured its activity in the hypoxia-sensitive tissues, 2 parts of the small intestine and in the lung. Our results have shown that the methane inhalation significantly decreased the xanthine oxidase activity in case of the duodenum and under the weaker hypoxia. In case of the ileum, the methane is able to decrease the xanthine oxidase activity significantly under both hypoxic circumstances. But in the lung, the methane did not exert significant effect on the xanthine oxidase activity. Nitrate and nitrate are stable markers of the nitric oxide. Consequence of their reduction, the nitric oxide production. And the nitric oxide have many advantages under hypoxic circumstances. The methane inhalation did not exert any effect on the nitrate-nitrate content in the duodenum. But in case of the ileum, the methane inhalation significantly increased the nitrate-nitrate content of the ileum under the strongest hypoxia. And in case of the lung, the methane inhalation almost significantly increased the nitrate-nitrate level under the stronger hypoxia. And we measured the nitrotyrosine level of the tissues as a marker of a nitrosative stress, so as a marker of a tissue damage. And under the weaker hypoxia, the methane inhalation significantly decreased the nitrotyrosine level of the lung, so may be able to decrease the tissue damage, but other data are under determination still, so we can't say sure consequence. The myeloperoxidase activity is a marker of a tissue damage of an oxidative stress. So, in case of the ileum, the methane inhalation significantly decreased the myeloperoxidase activity under the strongest hypoxia. Based on our results, conclusions are the following. Under hypoxemic hypoxia, xanthine oxidase activity does not change as during the well-known ischemia reperfusion. Methane significantly decreases the xanthine oxidase activity under hypoxic circumstances in the duodenum and ileum. And methane significantly increases the nitrate-nitrate levels of the ileum and almost significantly in the lung. In the background, the inhibition of nitrate reductase activity of xanthine oxidase is suspected. Methane decreases the nitrotyrosine level of the lung and myeloperoxidase activity of the ileum. So, all in all, the methane through decreased xanthine oxidase activity and nitrosative stress may be able to reduce the tissue damage, and with creating nitrate-nitrate pool for nitric oxide production, give an advantage to the tissues for healing. I would like to say a thank for my mentors, and thank you for your attention.
Dear Chairman, dear audience, our research work is focused on the smallest organic molecule, the methane. It's non-toxic and is thought to be biologically inactive. But these are not the reasons why our research group has started to work with this molecule. Methane can be produced by non-bacterial organisms, and the amount of methane in the breath can use for diagnostic purposes. In our research work, we are focused on the exogenous methane as a therapy. As my colleague has showed before, methane has a tissue protective effect in mesenteric ischemia and reperfusion. As you can see, this significant denudation of villi after 1 hour of 1 hour of ischemia with 3 hours of reperfusion. With methane treatment, the mucosa is almost as intact as sham-operated animals. So, what are the possible reasons of this incredible tissue damage during ischemia and reperfusion? It's oxidative and nitrosative stress, which biomarkers are hydrogen peroxide, superoxide, and nitric oxide. What's more, superoxide and nitric oxide forms peroxynitrite, which is an even more reactive agent. So these biomarkers lead to tissue damage. In our hypothesis, methane has a protein conformation modifying effect through which it influences the enzyme activity, and methane reacts with reactive nitrous species such as peroxynitrite. Our possible biochemical target is xanthine oxidoreductase. In normal oxygen tension, xanthine oxidoreductase oxidizes xanthine into uric acid while it releases hydrogen peroxide and superoxide. In the case of lack of oxygen and increasing reductive components such as NADH and in acidic conditions, xanthine oxidoreductase shows nitrite-nitrate reductase activity, and it produces nitric oxide. As you can see, xanthine oxidoreductase can produce the elements of oxidative and nitrosative stress in different amounts regarding to the circumstances. What's more, superoxide and nitric oxide can form peroxynitrite, that's even more reactive agent. Our aims were to examine the effects of methane on xanthine oxidoreductase activity and the xanthine oxidoreductase activity linked oxidative and nitrosative stress biomarkers and on nitrosative stress elements directly based on the peroxynitrite and methane reaction. Our method for the first hypothesis, so the enzyme activity modifying effect, we created a simple solution where we added the enzyme and nitrite and NADH, and we incubated our cuvettes at 37 Celsius degrees, and we bubbled it with normoxic air with or without methane for 10 minutes. After the reaction, we took out samples and we measured the kinetic xanthine oxidoreductase activity by fluorometry, the reactive oxygen species by chemiluminometry, and the nitrite-nitrate changes with photometry using Rees reaction. In our photometric measurements, we used an alternative pathway, and we used a substrate analog called pterine. In this reaction, we get a fluorometric measurable signal. Here you can see our results. We measured the xanthine oxide activity at 7.0 to 7.8 pH range. Methane decreased the xanthine oxidoreductase activity at every pH levels, and in 2 cases the difference proved to be significant with a paired t-test. The chemiluminometric measurements were to measure the amount of formed superoxide in our mixture. We used luciganin-enhanced chemiluminescence. And here you can see our results. As you can see, methane decreased the xanthine oxidoreductase-induced superoxide production, and at the pH level of 7.0, this difference proved to be significant. We measured the nitrite and nitrate content in our reaction mixture. So we could measure the nitrite concentration directly with grease reaction. But to measure the nitrate is a bit tricky. We had to use an enzymatic reaction to reduce nitrite into nitrate. And we could measure the diazonic salt. It's a colorful molecule, so we could measure it by photometry. In these figures, you can see the changes of nitrite and nitrate levels in our mixture, because we took out samples at the beginning of the reaction and at the end of the reaction, so we could calculate the changes during our reaction. The nitrate pool showed neither changes between air and methane treatment nor between groups. But in the case of nitrite, at the pH level of 7.0 with air treatment, xanthine oxidase shows nitrate reductase activity. Background of this nitrate consumption, we think that xanthine oxidase could produce nitric oxide. In the case of methane treatment, methane suspended this nitrate reductase activity of xanthine oxidoreductase. We created another method to measure the direct reaction of methane with peroxynitrite. We made a tyrosine amino acid solution, and we treat with 3 minutes of pre-treatment with the normoxicare with or without methane. Then we added the peroxynitrate, and after 2 minutes of reaction, we took out our samples and we immediately took them into liquid nitrogen to stop the reaction. We measured the nitrogen content of our solution, because this is the stable end product of peroxynitrate and the tyrosine reaction. As you can see, in the case of methane treatment at the pH level 7.4, methane could decrease the amount of formed nitrotyrosine. As a conclusion, we can say that exogenous methane decreased the xanthine oxidase activity and the reactive oxygen species production, and the nitrate reductase activity of xanthine oxidoreductase, which can possibly mean inhibition of nitric oxide formation. Methane also decreased nitrotyrosine production. We will need to do more studies to check whether there are physicochemical changes of our enzyme, and we had to specify this peroxynitrate and methane reaction, and this way we can measure the other nitrogen-containing molecules in our system. And finally, I would like to thank to the workers of Institute of Surgical Research and for you for your attention.